Hello friends, I am Shubhangi Rohatgi, your biology guide for today, and I welcome you all to the third and the final session of chapter 5, Morphology of the flowering plants. In the earlier session, we were studying the stamen. The androecium is made up of the stamens, and stamens have anthers and filaments. Each anther is bilobed, and in these lobes are present pollen sacs which produce pollen. What would happen if some anther fails to produce pollen? Anthers, or the stamens for that matter, are the male reproductive units of a flower. If the pollen grains are not produced, that means that particular anther or the stamen will be sterile. So, the sterile stamens are known as staminodes. There are many other descriptive terms also. Let us study them one by one. I have been emphasizing again and again in each of the classes of this chapter that learning to define properly, use of proper technical terms and definitions is very important when you describe a plant or an animal for that matter. The description should be in the scientific manner so that there are no confusions and the meaning is not lost in the conversation. So, as I told you, staminode, a sterile stamen is known as staminode. Epipetalous. Now, epi means over, petalous stands for petal. So, when the stamens are attached to petals, they are said to be epipetalous. The example is brinjal. Similarly, if the stamens are attached to tepals, that is the perianth instead of petals or corolla, the stamens will be known as epiphyllous. Phyll stands for the leaf, epi over, so over the leaf. Again, try to follow the logic instead of just cramming up the names. Epiphyllous stamens are found in lily. When there are many stamens and they are not joined together, the condition is said to be polyandrous. The androecium is said to be polyandrous when the stamens are free, but it is not always the case. It may so happen that the stamens join together to form a bunch. If only one bunch is present, then the condition is monoadelphous. So, the stamens here are joined together to form one bunch. Example is China rose. Similarly, if two bunches are present, then the androecium will be said to be diadelphous. Now, it is not necessary that in those two bunches equal number of stamens be present there. It may so happen that one bunch has only one single stamen while the other has many. And when more than two bunches are present, the androecium is said to be polyadelphous, that is having more than two bunches. Example is citrus. Now, in the earlier session also I had emphasized that do not focus on learning the examples. Try to observe as many flowers as you can from near your surroundings and then study their structure. Study the types of stamens present in them and decide on your own whether they are epipetalous, epiphyllous, polyandrous or monoadelphous, diadelphous or polyadelphous. To give you a taste of this exercise, let us do this thing. So, I have this flower here. This is a beautiful flower of China rose. Then there is another flower. This flower belongs to Cucurbitaceae family, and there is this beautiful flower of lily. Can you see all these three flowers? Now let us study them one by one. Let us begin with the flower of Cucurbitaceae family. Let me zoom in for you so that you can see the structure of the androecium. Can you see the androecium here? These are the stamens. These structures, they are the stamens. Now, please have a look that these stamens though appear to be very big, but are separate 1, 2, 3, 4 and 5. So, there are 5 separate stamens. What should this condition be known as? Yes, you are right. This is polyandrous condition. On the other hand, have a look at this flower. The stem is present here in the China rose. These are the stamens. Can you notice that they are all growing on one tube? They are all growing on one tube. So, this is which condition? Yes, you are right. This is the monoadelphous condition. Okay. Having talked about monoadelphous and polyandrous stamens, let us have a look at this flower, the third flower which I had shown you earlier. It needs to be zoomed out. Okay. So, let us try to dissect this flower. Let us open it. So, I am going to remove some of the petals so that we can see the internal structure of this flower. I will cut a vertical section of the flower. See here. By the way, have you noticed that this was the lily flower? And in earlier class we had discussed that this lily flower has a perianth instead of calyx and corolla. So, these white structures that you can see here are the tepals. And now look at the junction. Where are these stamens arising from? Yes, they are arising from the tepals. They are joined on the tepal. So, which condition is this? This is epiphyllous condition. Let me zoom in the specimen again for you. Have a look here. You can see that the stamens have arisen from the tepals. They are joined on the tepals here. So, this is an example of epiphyllous condition where the stamens are arising from the tepals. Now, let us see another example. I found this flower growing on the roadside. Let us dissect it open, cut it open and study its structure. It is a beautiful flower. Look here. Where are the stamens located? Can you see the stamens on this flower? Yes, they are located here. Where? On the petals. Why did I say that these are petals and not tepals? Because I can see the sepals here. If these are sepals, that means these are petals. So, these anthers here, stamens here are growing on the petal. So, these are epipetalous stamens. Clear? I would again request you to dissect open the flowers which are available easily to you. Find some locally growing weeds and some flowers. Try to cut them open, and if it is difficult for you to cut them open, you can simply break them open or tear them open. Look, I will show you how can you do that. Just remove one of the petals and slowly proceed on to tear it open, and you get a beautiful section of a flower. This is the LS, though crudely made, but it shows the LS of the flower. You can simply tear the flower open and study the internal structure and the arrangement. Here also, if you will see, this is the gynoecium. I will remove it for the sake of studies, and these beautiful stamens are located here on the petals. Okay, I am afraid you cannot see the stamens. Let me zoom in. Can you see the stamens? Now, these are the stamens bearing the anthers. Okay, let us move on. So, this was all about different types of stamens. I will also show you some pictures of epipetalous stamens. You have just seen stamens growing on the petals. These are epipetalous stamens. Now, here as I said, when the stamens are present in bunches, one bunch can have only one stamen, the other can have many. Such a condition is known as diadelphous androecium. And when the androecium is present as one whole bunch, the condition is said to be monoadelphous. Now, we will move on to the study of gynoecium. Gynoecium is the female whorl and it is composed of carpels. Let us draw a carpel. Let us zoom out so that you can see what am I drawing. Let me draw a carpel for you. A typical carpel has three organs: the stigma, the style and an ovary. The stigma, the style and an ovary. This part is the stigma. Can you tell me the function of the stigma? What does it do? Yes, the stigma forms the receptive surface to receive the pollen grains. The style forms the junction between the stigma and the ovary and takes the pollen tubes to the ovary. Inside the ovary, many or one ovule may be present depending upon the taxa. Let me draw an ovary with only one ovule. Such an structure you also studied in junior classes. So, this is the ovule present inside the ovary. Please remember this structure and these names when we study the gynoecium in detail later on. The stigma, the style, the ovary bears the ovule. After fertilization, these portions wither away and the ovule matures into the seeds while the ovary develops into a fruit. When only one carpel is present, the flower is said to be monocarpellary. This is a monocarpellary condition, but many a times more than one carpels are also present in a flower. Then there is polycarpellary condition. For example, here two carpels are present but they are not united. Such a condition is apocarpous condition, where many carpels are present but they are free, they are not united. For example, as in rose. Now, syncarpous condition is when many carpels are present but they are united. They may be united to various degrees, various extents. Such a condition is known as syncarpous condition. Here the carpels are united. For example, in tomato. You all must have seen the rose plant. Let me open a rose plant for you. So, what will I do? I will just remove the petals one by one carefully without harming the internal structures that we want to see. Let us zoom in. Okay. So, this beautiful flower is densely covered with the stamens, many stamens. We need to remove these stamens also because we are studying the gynoecium. So, let us remove the stamens also. Now, I have removed the stamens. Sorry for being a bit messy, but we are studying the flowers. We are allowed to be a bit messy. Okay. Now, here I have removed most of the stamens, not all, and can you see various little thread-like structures? What are these? These are many styles and stigma of the many carpels that are present inside the rose. We have just studied that rose has apocarpous condition. That means these styles are not fused. There are many carpels and they are not fused. That is why we can see there are so many styles and stigma. It might not be a very good specimen for you. The better specimen will be one when you will grab a flower of rose and try to open it up and look at the various arrangements of the stamens, look at the number of stamens it has and have a look at the apocarpous condition by seeing the stigma and the style which are lying freely. Now, we will move back to the diagram of the ovary that we had just made. We had talked that each ovary bears one or many ovules. Here is this diagram, and we had talked that each ovary bears one or many ovules, but ovules do not arise haphazardly inside the ovary. Well, nothing happens haphazardly for that matter in an organism. So, ovules arise only at the point where placenta is present. Placentation is the arrangement of ovules within the ovary. There are many different types of placentation that are possible. We will study them one by one. First one to come up is the marginal placentation. What happens in this case? The ovary is somewhat like this and a placenta develops on this suture. So, many ovules are born here in this manner. They appear to be arising on the margin of the ovary. Such a placentation is called marginal ovary and the example is P. From which structure can you really see the placentation? If I ask you to tell me please which kind of placentation is found in a tomato plant, what will you do? Will you go out and try to find a tomato flower, cut it open, cut the ovary open and then study the placentation? I will tell you an easier method. If we want to have an idea what kind of placentation did the ovary had, we can study the structure of the fruit because the fruit develops from the ovary itself. So, the placentation will also be present in the fruit which was present in the ovary and it might be present in the form of septa, and whether the placenta is present or not, but we can see the seeds which have developed from the ovule. So, the position of the seeds will resemble the position of the ovules which were present there. So, if I want to know which kind of placentation is present in the tomato, I can simply cut open a tomato fruit and study the placentation from that. So, the next type of placentation is axile placentation. What happens here? The placenta is axillary and there are many locules and the ovules are born on the placenta, so that many ovules are present in each locule. These chambers of the ovary are known as the locules. Have you ever seen such a fruit where you can see the axile placentation? Think of many different fruits that you cut open. Think of the fruit shapes when you cut them transversely, not longitudinally. This is the TS of ovary. So, think about the TS. Well, I will help you. What is this? Lady fingers, and here you can see the axile placentation. There are 5 locules here, and in each locule many ovules must have been present, but in TS you can see only 1 locule, right? So, this was axile placentation. Any other example that you can think of? Well, save it for a while. I will ask those examples later from you. Next type of placentation is parietal placentation, where the placenta is present on the periphery, so that the ovules arise from the periphery of the ovary. The ovary is unilocular, that is, it has only one chamber. Have you ever seen a fruit which shows parietal placentation or parietal arrangement of seeds? Think of the fruits that you can which show parietal placentation. Let me help you. Can you identify this picture? This is a section of a melon, and see, look how beautifully there is the resemblance between the placentation, the orientation and arrangement of ovules and arrangement of seeds in this picture of melon. Can you think of any other example which shows parietal placentation? You must have cut many fruits and vegetables many a times. Please try to notice the type of placentation that is present in them. The next one up is free central placentation. What happens here is that the placenta is present in the center and the ovules are born on the placenta, and there are no septa. Such placentation is found in many flowers like dianthus. The last one to come up is the basal placentation, which I had drawn here also. So, the ovary is unilocular, mostly only one ovule arises from the base, that is why the basal placentation, and when you cut the section you get something like this. Example is sunflower. So, these were the 5 different types of placentations. Now, I have a little exercise for you. I have got some sections. This is the section of a tomato. This is section of a capsicum, and this, can you tell me? Yes, it is a cucumber, some of the very common vegetables from your kitchen, and this is what I want you to do also. So, I have cut them open for you. I have cut a TS of these vegetables, and technically they are the fruits, and I want you to study the type of placentation present here. So, here in this tomato you can see there are these septa and there are these locules, the chambers of the ovary, and the seeds arising from this very placenta. Which kind of placentation is it? Can you answer? Yes, it is axile placentation. Similar placentation is also present in the capsicum. There are these locules and the placenta bearing the seeds. Now, this one is different. Here there are no locules and the placenta is present on the periphery. So, this is the parietal placentation. In this manner, you can study many different placentations. Let us move on. The next topic that we have to cover is the fruit. Now, when the fertilization takes place, the ovary develops into the fruit. Sometimes ovary develops into the fruit even without fertilization. Such fruits do not bear seeds and they are called as parthenocarpic fruits, but we are talking about the fruits that bear seed. So, if this was the carpel with the style, stigma and a swollen ovary, what will happen when the fertilization has taken place? These parts will wither away, okay, and suppose there was this placentation, free central placentation. Let me draw a free central placentation for you. Many ovules were present here. Now fertilization has occurred and these flowers have withered away. Now the ovary wall swells up. And this is how a fruit is formed. This structure will now be known as the fruit and these will be the seeds. This is the fruit wall. Now this fruit wall can be fleshy. This fruit wall is known as pericarp. It can be dry or it can be fleshy. When it is fleshy, it is divided into three portions: epicarp, mesocarp and the endocarp, starting from the most exterior layer, epicarp. Then below it is the mesocarp and last one to come up is the endocarp. Now depending upon the composition of three layers of the wall, we consume different layers of the fruits. For example, in mango, the epicarp is that covering the peel that you throw away for the herbivorous animals to eat, and mesocarp is the pulp that you consume. Endocarp is the innermost hard layer that is present inside the mango fruit. What about banana? You all must have eaten banana. Again here you peel away the epicarp, throw it off, and inside you find there is only one thing. Where is the mesocarp? Mesocarp is present in the banana in the form of the fibers that are attached to the endocarp. So if this is endocarp of the banana, please notice that there are some fibers also. These fibers are the mesocarp. So mesocarp and epicarp both are the edible portions of a banana, and you throw away the peel, which was the this was the endocarp and this is epicarp, and you throw away the epicarp in banana. What is a drupe? Drupe is a one-seeded fruit which has developed from monocarpic superior ovaries. Examples are coconut and mango. This is the picture given in your NCERT book showing the drupes. Here in coconut, the mesocarp is fibrous and we consume the seed itself. Coming over to the seeds. I have got some dicot seeds here. These are chana or the gram seeds. Now each seed has a seed coat which is made up of testa and tegmen. If you soak the seeds for a long enough time, you can easily remove the seed coat. To study the internal structure, I am removing seed coat of one of the seeds for you. Here I have removed the seed coat to show you the internal structure. This is the seed coat which I have removed. I have kept it, placed it here with the seed. This is the seed. Now inside the seed you can see, first on the seed, let us see what is present. We can see a mark over here. This mark is the hilum. This is the point of attachment of the seed to the fruit, and there is a pore like structure. This structure is the micropyle. This was the opening from where the pollen tube entered into the ovule. Now if I cut open this seed, what will I find? This is a dicot seed, so I must find two cotyledons. Seed also bears a radicle. Let us try to see the radicle also. Nice. So I have dissected this seed. Let me focus the structure for you. this is the dissected seed. Can you see the embryo here? Yes, and this is the radicle and this is the cotyledon which stores the food. For the sake of better understanding, let us have a look at the NCERT diagram. So, we studied about the micropyle, the hilum and the two cotyledons. The embryo gives rise to the radicle and the plumule. I have told you the functions of all these structures. Cotyledons store the reserve food material. Sometimes endosperm, which provides nourishment to the growing embryo, persists, while in others it is completely consumed. Now, in dicot seeds, this was the structure. Let us come over to the monocot seeds. In monocot seeds, there is only one cotyledon and mostly the endosperm is present. It is persistent, it is not consumed totally by the growing embryo. There is one layer which is known as eleuron layer. This eleuron layer is made up of proteins and it separates the endospermous part from the embryo. The embryo has one cotyledon and the name of the cotyledon is scutellum. The coleoptile covers the plumule of the embryo and coleorhiza covers the radicle of the embryo to provide the protection. This was all about the fruits, the seeds, and up till now we have studied almost all the parts of a flower. We began with the four whorls, we talked about position of the ovary, then we talked about the structure of the calyx, the corolla, the androecium and gynoecium with the variations that can be present in them. We also studied the structure of a typical fruit, a drupe, and then we studied the dicot seed and the monocot seed. So, almost all the parts of flower have been covered now. Now, let us learn to describe semi-technically the flowering plants. Now, since beginning I have been telling you that when we study morphology, it becomes very important that we learn to describe the features also. So, we have to learn to describe these features in a very scientific manner so that the description is apt, not very long, not very brief, but it should be accurate and correct all the time. And at the same time, we should learn to use some symbols and structures to convey the message clearly and easily. Just as in case of chemistry, you have learned symbols for so many elements. We have also developed some symbols to depict the various types of floral structures, and these symbols are used in the writing of floral formulae. Similarly, some structures have been developed to draw the flower easily. If you try to draw a flower every time you describe it, it will be very difficult and it will not be easily copied also. It will not be possible to draw same diagram again and again, even by a practiced artist. So, we have developed a way to draw floral diagram whereby we show the structures of the flower, the floral whorl, and we show their arrangements in the flower. We will learn about these things now. So, when we are describing a flowering plant, first of all we have to describe its habit, whether it is a climber, creeper, herb, shrub or a large tree. Then we talk about its vegetative character, whereby we talk about its roots, stems and leaves. Which kind of roots do they have, fibrous or taproot, the stem, the modifications that are present in the stem, the leaf, the shape of the leaves, the type of leaves, the phyllotaxy, everything is described in short. Then we come over to the floral characters. In the beginning, first of all, we describe the inflorescence, which kind of inflorescence is present, whether it is racemose or whether it is cymose. Then we come over to the description of the flower parts and we describe the four whorls one by one. And finally, this description is followed by a floral diagram and the floral formula. This seems lot, but when we study the flowers properly, study the plants properly, this becomes very easy to do. So, let us learn about the symbols for writing the floral formula. We begin with the presence or absence of bracts. If the bracts are present, I need to zoom it out so that you can see what am I writing. If the bracts are present, we use the term BR. This is for a bracteate flower. And if we write EBR, it is ebracteate flower, that is the bracts are absent. Then we have to tell whether the flower is actinomorphic or zygomorphic. Actinomorphic, if you remember, I told you, is the flower which shows radial symmetry. So, actinomorphic flowers are shown by this symbol, while the zygomorphic condition is shown by this symbol. These are simple symbols, and I am repeating, just as you have learned the symbols in chemistry, learn these symbols also, because these will help you to describe the flowers very easily, just as those symbols in chemistry help you to write the equations easily. The third thing that we focus or tell in a floral formula is that whether the flower is unisexual or bisexual. So, unisexual flowers can be either male or female, while the bisexual flowers are shown by this figure, this symbol. This is the symbol for bisexual condition. Then we talk about calyx. Calyx is shown by the symbol K. Now, writing only K will not serve the purpose. We also have to tell how many sepals are present there, whether there are 4 sepals, 5 sepals, 8 sepals or 10 sepals, we have to tell how many sepals are there. So, we write the number here. I am writing N to denote the number. Now, we also have to tell whether these sepals are united, that is fused, or free, and we can do so easily. If they are united, we put a bracket here. The bracket shows united condition, and if the bracket is not there, it shows that the sepals are free. For example, if I write K5, this means there are 5 sepals and they are not united. On the other hand, if I write K5 enclosed in a bracket, this means there are 5 sepals and they are all jointed. Similarly, I can also write K2 plus 2. What does that mean? It means that there are 4 sepals in total, but they are present in 2 rows of 2 each. Okay? The next one to come up is the corolla, and just as we had symbols for united corolla, calyx or separate calyx, we have symbols for united or free corolla also. Corolla is denoted by C, and the number of petals is written here below the C, and if I write the number in the bracket, that means these petals are fused or united, and if I write the number only without the bracket, that means the petals are free. So, C5 means 5 free petals, and C5 enclosed in a bracket means 5 fused petals. Again, C1 plus 2 plus 2, what does that mean? That the petals are arranged in 3 layers which are not united. The next thing is perianth. If the calyx and corolla are not there, then there will be perianth, and perianth will be shown by a capital P, again followed by the number and the bracket if required. So, this shows fused tepals and this shows free tepals. Lastly, we come over to androecium. Androecium is the male whorl, as you must have known by now, and it is depicted by A, followed by the number of stamens present. If these stamens are fused in a group, then we put the number in the bracket. If they are free, then we simply write A and the number. How will we depict the epipetalous condition of the anthers? We will write C followed by say 5 if there are 5 petals. We will write A followed by 5, suppose there are 5 anthers, and we will show a junction between the two. This shows that the anthers are growing on the corolla. Similarly, epiphyllous condition will be shown by this. Can you explain to me what I have written here? I am showing that this flower has a perianth instead of calyx and corolla, and there are 3 tepals, and the androecium has 3 stamens, and these 3 stamens are growing on the 3 tepals. It is simple if you try to follow the logic and the meaning of the symbols. Last one to come up is the ovary, the gynoecium. So, we show the gynoecium by a letter G. Now, if the ovary is superior, that means all the other structures are arising from below it. So, we put a line below G. While, so this is a superior ovary. But if the ovary is inferior, that means the other structures must be growing from above it. That is the epigynous condition. So, here by this symbol we mean that the ovary is inferior. Again, you do not have to cram up whether to put the line below the G or above the G. Just remember, what does this line depict? This line shows the origin of other structures, floral appendages. So, if the ovary is superior, the line will come below G, because this will make the ovary superior. If the ovary is inferior, that means the line should come over G and G should be below the line. Okay. We also show the number of carpels. If we write 3, that means there are 3 carpels present in the gynoecium, and if we enclose this 3 in a bracket, what will it mean? That these 3 carpels are fused. So, this is a syncarpous condition, while that will make it an apocarpous condition. We also write in the symbol locules and number of ovules per locule. So, this was all about the symbols of a floral formula. EBR for ebracteate, actinomorphic, zygomorphic, unisexual flowers, bisexual flowers, calyx depicted by K, corolla depicted by C, perianth by N, fusions depicted by closed brackets, and joints depicted by junctions, androecium by A, gynoecium by G. Now, let us try to write the floral formula of some flowers. I have taken an example of brassica. You might be asked to write the floral formulae of different flowers based on the description that is given here. So, let us start. We will also learn how to draw the diagram. So, let us first write the floral formula. The floral description begins that the flower is actinomorphic. So, what was the symbol for actinomorphic? This, right? Then it says the flower is bisexual. So, I will draw the symbol of bisexual. Then it also says the flower is hypogynous, but we write about the ovary in the end. So, just to remember, I will write G followed by the dash, that is, it makes the ovary superior. Now, there are 4 sepals arranged in 2 rows. Sepals that will be denoted by K, and there are 4 sepals arranged in 2 rows. So, it will be 2 plus 2 followed by 4 free petals in valvate estivation. Now estivation is not shown in the floral formula, but it will be used to make the floral diagram. So there are 4 free petals. Petals are denoted by the letter C, and we will write 4 here. We will not enclose them in the bracket. Why? Because they are free. Now it should be followed by the androecium. So for the androecium, it says here that there are 6 stamens diadelphous condition. That means they are present in 2 groups. So the androecium will be shown by letter A followed by 2 plus 4 diadelphous condition. Now last is the gynoecium. We will write the formula for gynoecium. It says that there are 2 united carpels. So carpels united and 2 with superior ovary and axile placentation. Again we cannot show the axile placentation in the floral formula. So this is our floral formula of Brassicaceae, and we can check it from here also. This is what we have drawn. This is what we have written: actinomorphic, bisexual, calyx in 2 rows of 2 each, corolla 4 free petals, androecium diadelphous 2 plus 4 condition, superior ovary, 2 fused locules. So you can easily decipher the floral formula also if you know the meanings of these symbols. Now we will try to draw the floral diagram for the very same flower. Okay, let us go back to the description. It said that there are 4 sepals in 2 rows. So we will draw the 4 sepals, 1 and 2. We will use the same technique that we had used while learning to draw the estivation. 3 and 4. So there are 4 sepals. We show the mother axis by drawing a dot over here. Okay, and it does not say anything about them overlapping each other. They are all free. So these are the 4 sepals. These 2 are outside and these 2 are inside. Right. Now we have to draw the 4 petals, that is the corolla. 1, 2, 3 and 4. This is how we have drawn the corolla here, 4 petals. Then the androecium with 6 stamens in such a manner that they are present in the order 2 plus 4. So I will draw 2 stamens here, 1 and 2 inside, 4 inside. What am I drawing? These are supposed to be the shapes of the anther. So we have drawn 6 stamens, and lastly we have to draw a superior ovary with 2 locules. So this is the ovary and these are the 2 bilocular condition, and this completes our floral diagram for the brassica. Let us compare it with the figure that is given in your books. The 4 sepals, the 4 corolla, the androecium and the gynoecium. You cannot cram this diagram, and in fact you do not need to cram it up because you can easily learn to draw it. We will further draw some other floral diagrams of some other flowers. Let us take an example of Fabaceae. Now Fabaceae is special. Earlier also when I was talking about the estivation, I told you that Fabaceae shows vexillary estivation. This is very typical of this family. We have to write the floral formula and the diagram. The flower is bracteate, so we will begin with EBR. Sorry, it is bracteate, so we will write BR. Then it is zygomorphic, so this symbol will be made. It is bisexual, so this symbol will be made, right? And the flower is hypogynous, so we will write G and underline it to show the superior ovary, but we will do it later. Now there are 5 united sepals, imbricate estivation. So sepals 5 and united. Estivation is not shown in the floral diagram. Then the petals are 5, polypetalous, papilionaceous flower. How is this papilionaceous flower arranged? 1 posterior standard, 2 laterals, 2 making a keel. Papilionaceous flowers have 1 standard, 2 laterals and 2 making a keel in this manner, and these are joined. This is the vexillary arrangement. We will come to this part later. So, but we can write in the floral formula 1 plus 2 plus 2 because they are present in 1, 2 and 3 layers. So, I will write C1 plus 2 plus 2. Stamens are 10 here. The stamens are 10 and the condition is diadelphous. So, I will write A 1 plus 9, right? And the ovary is monocarpellary, superior ovary, okay, unilocular, many ovules, marginal placentation. So, these are the flowers of Fabaceae family. You can see this flower is different from the flowers we have studied up till now. Here this is the standard, these are the 2 wings and these are the 2 keels, okay. That is why this shows the vexillary arrangement. Now the formula is BR, zygomorphic, bisexual, joint 5 sepals, vexillary arrangement of the corolla, 1 plus 9 arrangement. Yes, these 9 stamens are fused together, so we will put a bracket here, and the gynoecium is superior with only 1 locule. So, this is the floral formula here of the family Fabaceae. Please see here that the sepals have been made and these sepals have been jointed and the vexillary arrangement has been shown. Inside, the anthers have been shown in a diadelphous condition. Let us try to make this diagram. So, first of all we will draw the sepals, 5 sepals, 3, 4 and 5, okay, and they are supposed to be fused. So, first of all we will draw them. And then we will show the fusions. By this, right? Now we will show the arrangement of the calyx. There should be 1 standard which is going to cover the other 2. So, 2 laterals and finally 2 keel, and these keel are jointed. You do not need to draw these artistic structures always. You can simply draw the meaningful lines. Line diagrams will do for your floral formulae and the floral diagram. Now, this shows the vexillary arrangement. Now, we will show the androecium, which is made up of 10 stamens. For the sake of better understanding, I am using a different color here to show the 9 joint stamens. And 9. This is it. Now, after the androecium, what will we draw? We will draw the gynoecium and show the marginal presentation here. Show the mother axis with the dot, and your diagram for the family Fabaceae is complete. I am repeating again that you cannot cram up these diagrams, you have to learn to draw these, and you can draw these only when you have learned the meaning of the symbols. Similarly, we can write the floral formula and draw the diagram of family Solanaceae. Here the flower is actinomorphic. Let us do this quickly. Then it is bisexual. So, bisexual flowers are depicted by this symbol. The flower is hypogynous, ovary superior. 5 sepals are there. So, I will write K5 enclosed in a bracket, followed by 5 sepals. These are also united. 5 stamens are there, and now it says that this condition is epipetalous. So, I will show the epipetalous condition by this junction. Lastly, it says that there are 2 united carpels. So, and ovary is superior. There is axile placentation. We cannot show the axile placentation by the floral formula, but it can be shown in the floral diagram. So, this is the floral diagram given in your book, and this is the floral formula which we have just arrived at. Okay? Do not try to learn it, try to make it up on your own. Now, Liliaceae family. Here the flower is actinomorphic, bisexual, hypogynous, tepals 3 plus 3, stamens 3 plus 3. Let us see the structure. Yes, this is the floral formula of Liliaceae family. With this, we have finished our chapter, and let us now move on to the exercise questions. How will you justify that flower is a modified stem? If you have to frame the answer, please remember that the shoot apical meristem changes into floral meristem, and here the internodes do not elongate, and apex produces different lines of floral appendages at nodes. Similarly, you can try to define estivation and placentation if you follow up the chapter. How will you differentiate between apocarpus and syncarpus ovary? You write about the arrangement of carpels and give examples. Now, if you have to describe the arrangement of floral embers in relation to their insertion on thalamus, do not be confused by the typical language of the question. It is simply asking you to describe the hypogynous, perigynous and epigynous flowers with examples. Lastly, just as we have done so many examples here, please try to write the floral formula of an actinomorphic, bisexual, hypogynous flower with 5 united sepals, 5 free petals, 5 free stamens and 2 united carpels with superior ovary and axile presentation. If